I am really honored and excited to have Scott here with us today from sort of our local, you may have heard of it, Regeneron, certainly, you know, taking the world by storm, and I think a great example of what a small, good idea can turn into. And I'll save my mosquito quote for later. So, Scott, do you want a microphone? Are you good? You tell me.
I'll try to speak up. Can you hear me okay?
Can you move the microphone?
Sure.
Or you can
Yeah,
move it around like it's got a boom and the whole bit.
Well, one thing having a 12-year musician, I'm not daddy, I'm roadie, so I know how to run my wires and microphones.
Thank you. Thank you. That was so inspirational. That's terrific. Thank you to the organizers and thank you to all the earlier speakers. You've conveyed so many important things. And so I'm very pleased to be here with you and really excited about the initiative that you're doing, and hopefully we can participate. And so I was asked to talk about rare disease strategy at Regeneron. And just show of hands, who here knows Regeneron? Okay, so folks tend to know about Regeneron. Okay, so the company's been around for about 30 years, and I've been with the company now about 17. So it's a very different company since when I joined. There were about a little under 500 people. Now there's about 6,500 people worldwide, and we've had about 6 approved products. Me, I'm a rheumatologist and a scientist. Was training at Columbia University in the mid-'80s, doing a postdoctoral fellowship in immunology with Dr. George Yankopoulos, our chief scientific officer. And George, bona fide genius, went off to join Regeneron with Len Schleifer and start this company. And I went off and joined Pfizer and was at Pfizer for 11 years, worked with Jules, a colleague, and then after 11 years joined Regeneron. I developed my own rare disease thing about 12 years ago, a rare form of multiple sclerosis. So that's had its lessons within. So if we can move along, how do I change this?
Yeah. Okay, you got it.
Okay. So,
Do you want to point
I don't think I necessarily need a pointer, though it would help if you have one. Maybe I'll use it. Thanks. Okay, so, asked about the rare disease strategy at Regeneron, and was kind of thinking, why do we do rare disease research? Well, we do it for multiple reasons, and in part because we can, in large part because we learn, and also in large part because we must. First slide, next slide, please. Okay. So Regeneron's ethos and the tagline associated with the company is science to medicine. And we have this listing of sort of corporate expressions that characterize our culture, and number one is science drives our business and passion drives our science. So it's fundamentally all about science. And the core modus operandi of Regeneron really is to advance basic biological knowledge to discover key leverage points in pathways that are essential to health and disease. So fundamentally, we are a collection of biologists. We're a biology company, and we study important biological processes, look for areas of molecular levers, and then think about, well, where could this be applied? Okay? We work to continuously optimize platform technologies for discovery and therapeutics, as well as production. So discovery, like Jackson Labs, we do a lot of mouse work, make a lot of mouse models and develop methods for creating models very rapidly, but also other discovery platforms such as large-scale human genomics. I'm going to talk about that in a moment, the Regeneron Genetics Center. Platforms for therapeutics have included traps, which are receptor antibody fusion proteins. We're working on other therapeutic platforms now, such as gene therapy and gene editing, as well as platforms for production, ways to make more protein more efficiently with better success. But fundamentally, scientifically, we follow the science, and we will develop a molecule clinically whenever we believe there's an opportunity to make a difference for patients. And that could be rare diseases or it could be prevalent diseases. But rare diseases really teach us so much. The company is therapeutically agnostic. We're not married to cardiovascular or to oncology. We do everything. And if the experimental result says if you modulate this or that protein, it's going to treat atopic dermatitis, well, we'll go there. Or if it's going to treat a rare disease, we'll go there. Okay, next slide, please. So, we learn a lot from rare diseases. These rare diseases, and often Mendelian diseases, 80% of these diseases are Mendelian, they have critical human impact. They teach powerful lessons in biology. And we work to drive scientific progress through human genetics and genomics, through functional genomics in the mouse, through cell and molecular biology, and bioinformatics. My colleague Justin is going to tell us a good bit about these latter two elements. And in a moment I'll talk about genetics and genomics. We heard great information about functional genomics and mouse models of disease earlier. Next, please. Okay, so this is, Regeneron in general, overall as a company, try and discover and validate targets, find biomarkers that will help inform clinical development, apply these to traps and, try and develop therapeutics using cytokine traps and using antibodies, and then naturally produce the therapeutics and get them out there on the market. Okay, next please. Okay, a little bit about the Regeneron Genetics Center. And this relates a lot to rare disease. So about 5 or 6 years ago, George said, guys, that's his expression, hey guys, we gotta do this. Human genetics is really doing a lot to inform the discovery of targets for developing drugs. We need to start doing this at large scale. So we started learning about the world of human genetics, and next generation sequencing was just coming online and becoming a thing. Ultimately, at the end of the day, we decided, okay, we're going to do this ourselves. We were very inspired by the company Decode Genetics, based in Iceland, where they had their founder had this amazing vision. If we characterize the genetic variability of our population and map that to medical phenotypes within our population, we can learn a lot about disease. So they were doing SNP chips on 100,000 people, and they were beginning to sequence people and thought this was great. And, we were thinking about an alliance with them, and Amgen swooped in and bought them for way more money than we ever could have bought them for. And so we scratched our heads and said, well, we can do this ourselves. So we set up the Regeneron Genetics Center to do human genomic sequencing at scale. And we had a few foundational collaborations. 2 of them were in the rare Mendelian disease space. One of them was the Clinic for Special Children in Lancaster, Pennsylvania, and the other was Columbia University with Dr. Wendy Chung. And so a lot of patients with unsolved disease, let's do a lot of sequencing. Geisinger was large-scale population based. So the RGC had a fleet of 20 of these high-seqs. Now it's got 4 to 5 NovaSeqs, and is sequencing on the order of 4,000 exomes per week. So that's about 200,000 people having their genomes characterized. Not whole genome sequencing, exome sequencing. And all the information is stored up in the cloud. Next, please. So far, we've sequenced 300,000 exomes, and over 15,000 of these are for rare Mendelian diseases. So that's a big chunk of what we do. There's over 60 collaborations around the country and the world. This is fascinating, but I don't really have time to get into it too much more. But next slide. Okay, so really that's a general overview of Regeneron. Focused on science, therapeutically agnostic. Rare disease teaches us powerful lessons. And one of those lessons, Jules alluded to, we worked together on Regeneron's first project. So rare diseases teach us about biology, but they're also an important testing ground when you think you have a molecular lever that might work. And so now the Tour de France is running. And if you want to know if you have a bicyclist who can climb a hill, you go to Mount Ventoux. And in the case of, if you have a molecule and you think it might be a good IL-1 inhibitor, well, test it in Muckle-Wells syndrome, a genetic disease of overdrive of interleukin-1. And basically, that's what we did. We worked together and we found that this rare disease is just driven by this one molecular pathway, and if you have a targeted therapeutic, if it doesn't work there, it's not going to work anywhere. And basically, we started our study at the NIH, and on day one, we saw dramatic effects, and when Schleifer says, well, we're going to develop a drug for this indication right now, ultimately that became Arcalyst for the cryopyrin-associated periodic syndromes. In any case, I'd like to turn over to Justin, who'll give a case example of how one kind of works through a disease and pulls together information from a variety of sources to validate a therapeutic target. Okay. Justin, I'll buzz out of your way and I'll come back in a minute.
Right, so thanks everyone. I'm Justin Grindley. I'm in the Target Information Group at Regeneron, and for the bulk of my presentation today, I'm going to sort of run through a little bit of a case study, illustrate some of the work that we do, starting with genetics and then moving into modeling. Really sort of picking up on some of Scott's ideas that we're following the science and that we believe that human genetics can be a very powerful entry point into biology. So the topic that I'm going to tell you about today is an example of an extreme phenotype. And as Scott mentioned, rare diseases can be very important for sources of extreme phenotypes. But really, it's the severity of the disease that is capturing our interest in this condition, which is keloid scarring. So keloids are a disfiguring dermal proliferative disorder. They arise from aberrant healing of skin trauma. That can be lacerations, burns, surgery. As seen here with a 4-year-old child who suffered a burn injury. And these keloid scars are these sort of fibrotic nodules that you see here, particularly on the ear, but also on other sites. And the characteristic of keloids is that they extend beyond the borders of the original wound. And as you can appreciate, these dramatic scarring can interfere with bodily functions, and it can also have significant quality of life issues. There are rather limited treatment options for this disorder. Typically things like surgery, but also corticosteroids, sometimes radiation. And one of the challenges for that is that there's a high tendency to recur. So, for instance, surgically removing these lesions can sadly just lead to more scarring and more keloids. So we're interested in understanding this extreme phenotype because we think it's going to provide insights and therapies not only for potentially for these patients, but perhaps for other fibrotic diseases, whether rare or prevalent. But when we think about it, the process of drug development, we usually think of it as sort of divided into these type stages, perhaps selecting targets. That could be from lots of sources, literature, human genetics, animal models, and so on. If it's an antibody program, we would have a campaign where we discover and prioritize leads, perhaps based on their biophysical properties, things like affinity and selectivity. We'd then want to move on to some pharmacology, in vitro and in vivo, and of course then towards safety studies and so on. But as we look at keloids, what our scientists have recognized is that there's critical gaps in the knowledge of the biology. And I've highlighted where those gaps are here. One of the main gaps is the paucity of well-validated therapeutic targets, particularly genetically validated therapeutic targets. So one of the things we want to do is fill this gap. But then, you think we could do this. We've done it for lots and lots of targets. But what do we do here? Because keloid is a disease that is disrupting normal tissue architecture. So if we're just growing some fibroblasts here in a dish, in a monolayer, we're missing that normal tissue architecture of the skin, and consequently missing the aberrant architecture seen in the disease. So we don't think that these traditional monolayer culture systems replicate the structures that are disrupted in disease, so we need to look beyond those. And equally, there's no accepted animal model for keloid disease. So how are we going to do these experiments? So these are the gaps that our scientists in the fibrosis team are aiming to fill. And starting at the first of these, the human genetics, just want to sort of run through the concept that we have generally for applying the human genetics to fibrotic disease. So these will all be run through the Regeneron RGC, or Regeneron Genetics Center, that Scott introduced you to. And then the concepts there would be really to try and utilize human genetics to generate hypotheses. Things like novel disease-associated variants might give us insights into genes or pathways that might be disrupted. I'm going to show you in a moment how we apply it for keloids, but we can also think of this as applying more generally to many fibrotic diseases or diseases of the skin, including rare disease. But having these hypotheses is only the first step. So we want to not only have, say, disease-associated variants, but act on that information. And so with the idea that our goal here is novel disease models and drug targets. So that's where we turn to the Velocigene group, who were able to rapidly manipulate the genome in vitro or in vivo. So to generate things, for instance, like iPS, cell lines, these are induced pluripotent stem cell lines that you can then differentiate into the tissues of interest. And of course you want to compare them to the original human disease to understand how closely they're replicating the phenotype you're interested in. Going in vivo, we'd typically be generating knockout or knock-in mice, which will then pass to phenotypic characterization. We've heard a lot about phenotypic characterization today from the Jackson lab. But just to briefly outline what our process might look like, we'd be interested in developmental and adult phenotypes. In the case of these fibrotic disorders, we'd be interested in extracellular matrix structure. And for multifactorial disease, and this is definitely can be placed in that class, we think that we would want to look at things like response to stress. So in the case of keloid, that might be looking for it, for instance, a response to injury. And we'd be doing things routinely, such as gene expression profiles, that can give us molecular insights into what is the effect of the genetic change that we have made. So here's how that process has been working for looking at keloids. And the RGC projects, as Scott mentioned, we have many partners. Those projects can take many forms. But very much a theme is they're often done as a collaboration, partnering with groups who have access to cohorts of patients. And in this case, the partner was Ernst Reichenberger at UConn Health, and he'd assembled a cohort, a familial keloid from an African population. So he had 559 individuals in this cohort, including 28 families with at least 4 affected individuals segregating keloid. I'm going to massively simplify the work at the Regeneron Genetics Center, but there's a lot of tasks involved, from things like sample management, and whole exome sequencing, to variant calling, through to identifying disease-associated variants. And I also want to stress that this is a two-way process. So that we are, in particular, sharing the results back to the collaboration partner, which allows them to advance their own research, as well as to identify additional areas for collaboration. And in this case, the RGC was able to identify a variety of disease-associated variants. But I'm just showing you one of them. And this is a gene that I'll call gene X, and we'll show it segregating, variants in this gene segregating here with keloid in this multi-generational family. And in this case, we also had a separate variation in gene X that was segregating with keloid in another family in the same cohort, suggestive that these may be causative mutations that we're looking at in gene X. So this is the point that the target information group, the group I'm working in, get involved, and we dig into the biology, not only of gene X and its variants, its partners, but also how does that relate to the disease. And there's a lot of work there too, but I just want to give you sort of the bottom line of that, which was that they could recognize that gene X was a plausible keloid susceptibility gene. In particular, it had functions that are consistent with a role in skin homeostasis. And we see other examples of loss of function mutations that had a thick skin phenotype. And we also looked, although I won't go into a lot of detail about that, but possible relation to potential therapeutic targets. And I'm just going to give one example of such a relationship that we can recognize here. And all of this gives us additional motivation to go on and look at in vitro and in vivo modeling. So, as I mentioned, the next challenge for this area of biology is, well, what are your models going to be? And so, for that, we recognized that we wanted to get a more realistic skin architecture to be able to evaluate the effect of our potential therapeutics. And so we've teamed up with a company, MadTech, who have a lot of experience in creating what they call full thickness skin equivalents that they call the epidermal full thickness model. And this is what it looks like using a combination of normal fibroblasts and keratinocytes. But if we substitute in keloid fibroblasts, we get a slightly different looking structure. It's also more contractile, it produces more matrix, and we see alterations here in collagen gene expression, fibronectin, markers of activated fibroblasts like this, and also growth factors, CTGF, for example, that are implicated in fibrotic disease. So it's recapitulating some of the characteristics that we see in keloid lesions. So I told you we're interested in finding targets genetically, but that's not the only way we do it. We're also looking, for example, we're interested in the cellular basis of the disease, see if we can identify cells that have a particularly active role, and the signals. So one of the things that we're looking at is single cell gene expression analysis, where we generate single cell suspensions and then barcode each of those cells to do RNA-seq on them, so that we can not only identify what genes are altered in their expression, but which cells are producing those genes, that gene expression. And another next step would be more in vitro modeling. So now looking at the performance of those disease-associated variants, what do they do in the model, as well as using additional samples derived from patients. And some ongoing work that I won't show you in mice today, but we're engineering models with the keloid-associated gene variants, and of course, then we'll be interested in phenotyping them. So that just sort of perhaps gives you a flavor of how, from identifying extreme phenotypes that we think can be particularly informative, we take a combination of approaches, and so basically following where that takes us. If there's gaps in knowledge that we identify, we try filling those gaps. And if we'll be able to see this at the back of the room, but for the next couple of slides, I just want to change tack a little bit and pick up on the theme that Scott highlighted about how Regeneron is constantly looking to innovate its technologies and platforms, and particularly here, thinking about our therapeutic platforms. And for someone who, like me, who works in looking at disease biology and looking for targets, this is very exciting because effectively the space that we can consider a target just keeps on expanding. And as Scott mentioned, our first marketed product, ARCLIST, for a rare disease, the Cryopyrin-associated periodic syndromes, or CAPS, was a track. So it's a receptor extracellular domain fusion protein that captures excess IL-1 to alleviate symptoms. And we've had a lot of additional success with other traps, particularly for rare gel. But it's really the engineering mice to be able to produce fully human monoclonal antibodies that really opened up the doors for us to be able to target many other different kinds of molecules. And just a few of the examples here of what we're doing in rare disease illustrates how that process. So in things like familial hypercholesterolemia, both in heterozygous and homozygous forms, we have either marketed products or investigational products. We also have antibodies in clinical development for, again, very rare, very extreme diseases, such as fibrodysplasia ossificans progressiva, or FOP. I think we also see examples where we start with a prevalent disease, and as we did with sorilimab, which is improved for rheumatoid arthritis, but then follow up by looking at, well, what can we do with this molecule in a rare disease such as the juvenile idiopathic arthritis. And equally, we're going to have settings where the biology is pointing us as we should start perhaps our first clinical programs in a rare setting, as we're doing with our complement C5 antibody against paroxysmal nocturnal hemoglobinuria. So we're doing a lot with antibodies, and actually, if we consider this the sort of antibody space, we're continuing to do more and more with that. But we've also generated new technologies that are allowing us to do different things. So one of those is, for example, is antibody drug conjugates. So we can consider delivering drugs into cells using antibodies. And another is bispecifics, where we can do things like target two molecules and bring them together. Or as in this case, for B-cell malignancies, we can bring two cells together and use T cells to tackle tumors. And the first of these are just entering the clinic at the moment, and behind that we have additional technologies such as viral vectors, CAR T cells, and gene editing as a collaboration with Intellia. So, as we prepare for having these new technologies, and we really need to be to make sure that we have both the knowledge and the systems in place to really get the best out of their potential. And so, for my last slide, I just wanted to sort of give you an example of some of the informatics efforts that we do to support the target discovery programs. And so, as an example, enzyme replacement therapy, which we're very interested in, and antibody drug conjugates, which I mentioned, both require better strategies for cell and tissue targeting. They're both trying to get a molecule into a particular cell. And so the approach that we're taking is trying to find ways that we can combine lots of different types of data, some with tens of thousands of molecules, some perhaps with hundreds, some from smaller scale studies that we've done on individual molecules, and make that easy for scientists to add data, to search it, and to share it, so that we can also get to sort of integrate across the company, and people can learn from each other. So, just as my last slide, I want to thank all the people who've been involved in this project. This was led, the fibrosis teamwork was led by Laurie Morton's group, with Scott MacDonald taking a key role. I'd need a whole extra slide to thank the RDC. I'm going to highlight Claudia from the Translational Genetics Group. Of course, all the genetics work was a collaboration with Ernst Reichenberger, and the modelling work is a collaboration with MapTech. And within the Target Information Group, Hong Zhang has played a vital role in bringing all that data together for the
Thanks,
next technologies.
Thank you. Justin, would you, if you could move that boom around, maybe hold it. Thanks all. I'm just going to spend a few minutes Justin, would you be kind enough to work the slides for me?
Sure, yes. Here's the pointer too.
All right, thanks. Okay, so I'd like to take a little moment to talk a bit about clinical development. And so many of the themes that were mentioned today, I think I'm going to reiterate them. You've said them all. But basically, a couple of years ago, I was tasked with looking at what are best practices in the world of drug development for rare diseases. And I spoke with colleagues across the industry in many different companies, in academia, in government settings, as well as in the patient advocacy universe. And everyone really was just so kind and helpful. And so kind of put things together in what I call a bit of a framework for rare disease drug development. Just briefly, I'm amazed we haven't seen this slide today, but I thought we'd see it a few times before I got here. But just very briefly, really rare diseases in aggregate are not that rare. They're pretty common. Almost 10% of the population worldwide has a rare disease, and that means about 30 million Americans. We all know people with rare diseases. About half those diseases affect children. Many of them are devastating. Essentially, all pediatric cancers are rare diseases. 95% of rare diseases have no treatment available. About 5% do, and those rare diseases are not necessarily orphan. There can be a fair bit of activity in some of those settings, and that's a whole nother story about how do you develop a drug when there's 5 companies trying to chase after 200 people. And rare diseases actually represent 40% of recent FDA approvals. It's a very interesting world. Okay, next slide, please. The Orphan Drug Act was mentioned earlier, and that's been hugely important. At the time when the Orphan Drug Act was being passed, literally only 2 drugs were approved that year for rare condition. And you can just see over time, now there's about 40 to 50 drugs a year being approved for rare disease. So this is a hugely important piece of legislation and policy. Create incentives so people will invest in developing medicines for very, very small populations. You know, on that first slide it had said a rare disease is defined as less than 200,000. But 85% of all the rare diseases, and there's over 7,000 different rare diseases, the vast majority are Mendelian diseases, where the numbers may range from a handful of people, maybe up to cystic fibrosis has 28,000 in the US. So, you know, most rare diseases are around 1,000 or 2 in the US. Very uncommon. Next slide, please. So Regeneron's very active in the world of rare disease. That's an example of a person who was exposed to a little cold and developed this systemic inflammation due to overproduction of interleukin-1. And this is a condition we're working on right now. It's called fibrodysplasia ossificans progressiva. which means fibrous tissue grows abnormally, it ossifies, it turns into bone, and it does this progressively. It is a terrible condition. But again, going back to really advancing the science, our scientific colleague, Harris Economides, was working on this in the lab. He built a mouse model. He was doing cell and molecular biology. He discovered a totally unexpected aspect of the biology of this disease, which showed that in the setting of the mutation that causes FOP, a protein that's normally part of the paracrine system for communicating between cells acts as a stimulator to stem cells, and instead of making more muscle, it sends them down a wrong path to make bone. And we had a monoclonal antibody on the shelf already, directed against that protein, so now that's in a phase 2 clinical trial. We mentioned some of the diseases. Every one of our therapeutic areas that are doing research, they're working on some rare diseases. Next, please. So, doing clinical development, how do you take something out of the laboratory and bring it into the clinic? Well, said there's over 7,000 rare diseases, and each of them has what you might call an ecosystem associated with them. At the center of that ecosystem is the patient, and the family, and their doctor, and other people. So there's the community of patients, there's the community of academicians, there's the community of regulators, there's what companies are working on it, what scientists are working on it. So it takes a village, you know, and 7,000 diseases, every one of them is different, and every one of them is at a different stage of evolution and organization. You can have the Cystic Fibrosis Foundation, which is incredibly well organized, with clinics all over the country and with $3 billion in the bank. Or you can have individual people just looking around going, where can I find someone else who has this disease? How are we ever going to get anyone to study? So these ecosystems are characterized by scarcity and abundance. We certainly heard a lot of passion from Michael. There's an abundance of passion and drive for cures. Everyone wants to help. We heard earlier the regulators are abundantly passionate about making it happen. Orphan Drug Act, the incentives, the vouchers, the tax breaks. That's all good. And what we need to do is leverage that abundance to overcome the scarcity. Fundamentally, we don't know a lot about these diseases. Patients are few and far between. There's little drug development precedent. There are no endpoints. Nobody knows, what do you really study for this disease if you want to get a drug approved? So, how do we deal with that scarcity? Next slide. Well, basically, and we heard about this, we need to engage very intensively. We engage with advocates, with patients, with parents, with researchers, and with regulators. All of them need to work together. We need to do additional learning in the clinical setting that goes beyond the clinical drug trial. You can't just have a drug and do a drug trial. You have to know what to study. So you do natural history studies. You characterize the genetics of the population to find the right subpopulation. You listen to the voice of the patient, of the parent, of the caregiver. You've got to understand, you can't just develop a drug to change a number on a chart. You've got to do something meaningful. I mean, the reason you're doing it is you want to do something meaningful for the patient. So you want to hear what they need and what they want. In order to develop what we call patient-reported outcomes and endpoints for drug registration. And then one needs to be creative. You need to think outside the box. You can't do the standard 2,000 patient parallel group clinical trial. You may only have 15 patients to work with. You may need to use historical controls. You may need to put patients through multiple cycles of intervention. So one needs to be creative. And as mentioned earlier, you need to figure out where the patients are. You need to find patients first. Prospective patient identification. Sometimes this comes together if you do natural history studies. So all these things work together in order to overcome scarcity. Next slide, please. So in summary, rare disease research and development addresses important human needs. It teaches us about biology and about pathophysiology. And it opens doors to new modalities of treatment. Sometimes your treatment may be great and it's wonderful, you're treating that rare disease. Sometimes you find out it's not just affecting inflammation in cryopyrin-associated periodic syndromes, but it's preventing inflammation in the lining of blood vessels, and it can be used for atherosclerosis. That's something that's happening in the world. IL-1 is pioneered by Novartis with their anti-IL-1 drug. So, in summary, next slide, please. I just want to thank you for your attention. I know we went well, and really with appreciation to all of our colleagues in company and academia and industry, and most importantly to the people living with rare diseases who volunteer in research to try and improve health for all. Thank you for your attention.